So good morning, everyone. I hope you had a nice dinner yesterday. And I just thought it was cool to mention that I got interested in oscillations when I had classes in 2007 during my master's from Ole and Marcel. And so it's actually very special for me to be able to give a talk at the same event as they were giving. And there are two other things that I wanted to mention as well. So I'm a language production researcher, and I thought it was, let's say, intriguing, the fact that during those 2 past days, I may have heard the word speaker maybe 3 or 4 times. And I keep thinking, like, if there's a listener, it's only because there's a speaker. And so where's the speaker? So I'll be the only one talking about production, and I won't be talking at the same level that most of you were. But maybe sometimes it could help also that you think, in trying to understand some of the things that you're trying to understand, maybe there's something to be sought in the constraints within the speaker, who knows. And the other thing is that, I don't know if Jonas is here. He made it very hard for anyone coming after him, I think. So I had planned my talk to be kind of speculative and hopefully a bit provocative, and then after his talk yesterday, I thought, oh no, I have to change this entirely. But then during the discussion, I got encouraged again. I said, no, I'll just do it the way that I was planning on doing still. So what I've been trying to do, and the way that I've been trying to think in the past years, working on language production, is that, especially for language production, or not especially, but in general, language evolved once we already had a memory system. And for language production in particular, we also had already motor stuff going on, right? So to me, it's very plausible that whatever mechanisms were in place at that time are now being used to subserve language processes. And that's the idea that I want to test with you, that maybe oscillations are the best way we have at the moment to test where these things come together, like what are the fundamental mechanisms that are enabling us to speak and retrieve words from memory in the language domain. They're actually also used in other domains outside of language, right? So that's kind of the idea that I'm working with. And for that, and this is something that Jonas would probably say, that's all wrong, but for that, I've been using these signatures, and actually I know I can go there. I'll be using these signatures for now and saying, like, I think you're familiar with most of them. So theta power increases in mid-frontal areas, medial frontal areas have been often associated with cognitive control. Theta power increases in the medial temporal lobe with memory. Alpha beta power decreases in the lateral temporal cortex with memory. I was hoping Simon would have given his talk yesterday, and then it would make more sense for me. And beta power decreases in motor areas in the motor domain, right? So these are the signatures that I'm kind of relying on to be able to, or to see if I can understand some of the things that I'm seeing in language paradigms in relation to these other domains. So for my talk to make sense, I have to have this first part in here about the medial frontal theta, but given the discussion yesterday, I'll focus mostly on the second part of the talk, so I'll go very fast over this first part, and I hope not too fast. I'll show you more than Hufan did for his wheel wagon illusion. So I'm calling these conflict tasks. I think you know caller word Stroop, the Simon task, and this is an example of this task. If you look at the difference between a condition where there's more, let's say, conflict versus a condition where there's less conflict, you find this these theta power increases, and they are usually very much medial if you localize these sources, ACC-like, and with EEG it looks like this over the scalp. So it's the theta band, and it's around these time points here. So there's more theta power around 300, 250 to 500 milliseconds, something like that, when there's more need for control because there's more conflict relative to when there's less. That's the idea, at least. So this is for the Simon task. I think you're also familiar with the Stroop task. I don't need to say much about this. So you have to name the color of these words. And oh, now I see that I have this wrong. Sorry about it. This should be neutral because it's not a color name. And this should be incongruent because you have to say red even though the color is blue. or the word in there is blue. And this is, again, work by Simon. And I'm sorry, the plots look very different. He turned it upside down. But basically here what he's showing is that, again, there's theta power increases around this 300 to 500 to 700. It's always around that interval. And there's more power for the incongruent condition. And if you look at a source in the ACC, you see that there's more power for the incongruent condition than the neutral, than the congruent condition, right? So this seems quite clear across tasks that that's kind of how it works. So I'm using this schematic way of showing you this. So Stroop theta, more for incongruent than neutral than congruent. And that's also what you find in the reaction times. Now, for speaking, I think by now most language production researchers agree that we need some type of control process to regulate, monitor the things that we are doing when we are speaking. And so that's most of what I did during my PhD. And the idea is, can we understand a bit more what these control mechanisms are? And in particular, are they domain general or are they something specific to language? So a way that we have to test this need for control during production is by using picture-word interference. So people have to name the picture here in all these three conditions, and there's a distractor word on top that has to be ignored. And in this case, because they are semantically related to each other, they keep priming each other, so it's harder to resolve the competition in this case relative to the neutral case, and this is the easiest, of course, because they converge on the same representation. So the amount of control required is the largest for the incongruent condition. And that's also what you find in the reaction times. People are slower when the competition is harder to resolve. If we look at the difference between these conditions, what I'm showing you here, this is MEG data. The difference between the incongruent and congruent and incongruent and neutral. And what we see are these theta power increases that they are kind of frontal, left lateralized more or less. So it's again in the same range and in the same frequency range and time range as what we find for the Stroop task and the Simon task. And if you localize those sources, it ends up somewhere kind of, I projected everything to the surface here, so it could be that it's coming from the ACC, but here it looks, at least over the surface, like superior frontal gyrus. And if you take the TFR of this source here and there, you see again that there are these power increases, theta power increases for the incongruent condition relative to these other conditions that require less control, let's say. So both reaction times and the theta seems to follow this need for control. So that seems to be similar to the rest of the literature working on these conflict tasks. And this is work that is still in progress, but I thought it holds also across some patient populations, it seems. So this is a bit different. Now there's a neutral word versus XXX, which is even easier because there's just no lexical competition going on. And the need for control follows this particular line. And these are brain tumor patients the day before the resection. So they go undergo an MEG session to determine the dominance, which hemisphere is dominant for language, and we are trying to find tasks that were a bit more sensitive than just picture naming, because there's more to speaking than just picture naming. And this was one attempt to find a task that would be a bit more sensitive to tell us something about recovery and so on. So I'll just show you quickly. This is the difference between the two conditions. So people are about 55 milliseconds faster when there's no lexical competition relative to when there is lexical competition. And ignore these crosshairs, but if you look at the source of an effect that happens in the theta range between, in this case, 300 and 500 milliseconds, both stimulus locked and response locked, a source in the ACC that shows these theta power increases for when there's more need for control to resolve competition relative to when there's less. So theta and NRT again follow the need for control. So the field of language production, I think, is okay with the idea that lexical selection is regulated by frontal mechanisms. These, from my work, seem to be associated with this midline frontal theta. And so this potentially would allow me to argue that speaking also engages these domain general control mechanisms. So there's nothing really language specific to it. And this is now the paradigm that I'll spend most time on, because I think it's also related to what Marcel and Ole discussed yesterday. I don't see it necessarily as a prediction paradigm, but you may even see it under that frame as well. So it's still picture naming, but here I was interested in, when we speak, it's not always a picture is shown and then I speak. There's something going on, and that's how words come from when we are speaking. So they are internally generated given a particular context that we are in. That's what I was trying to do with this task. Have people name pictures, but they were preceded by a sentence that would give you some context. And that last word of the sentence was removed and then presented as a picture that people had to name, okay? So they either hear, she locked the door with the, or she walked in here with the. And what you can see is that from she locked the door with the, you almost get key for free, right? It just comes. Whereas she walked in here with the, it could end in any way. So there's no really constraint in this type of a sentence. So I'll be comparing these two sentences for everything that you'll see. And another thing that I wanted to mention, everything that I'll show you is based on cluster-based permutation. At the Donders, we are not allowed to use anything else. It's just not true, but everything I'll show you is that. So in this first study, I was in Berkeley with Bob Knight, and so these are patients, we discussed a bit of that before already. These are patients who are undergoing monitoring for epilepsy, so they are implanted with intracranial electrodes, and these are stereotactic, so they are coming laterally and then targeting the hippocampus. And so I had 12 patients do this task, and here are the reaction times for a constrained versus unconstrained context. So this is the picture naming reaction time, which I count from here, right? So once they see the picture, they name it. And you can see the data for every patient. Everyone shows that slope. This effect is significant within every patient over trials, so it's a very robust effect. And this is just to give you an idea of what the coverage was. I'm not sure you can really see it very well, but we have left and right coverage, and some of the electrodes are not really in the hippocampus, but they are more, some are in the parahippocampal gyrus and entorhinal cortex and so on. So what you see here is the difference between the two contexts, and these are the t values from the cluster-based permutation so that's the statistics between these two conditions, and everything that is not significant I just masked. So what you see here is what the results of the cluster-based permutation. And for 10 out of the 12 patients, what we find are these theta power increases during the course of the sentence. So this is time locked to the picture. During the course of the sentence, there's theta power increases when there's more context relative to when there's less context. And I'm taking here even, I think, something, I'm not sure these patients, anything between 2 and 10 hertz to be the theta rhythm in the hippocampus. Joshua Jacobs has a paper on that saying that that's for the human hippocampus, that's the range that we can still call theta between 2 and 10. So I'm interpreting all of these power increases as theta power increases in the medial temporal lobe when there's more context relative to when there's less context. And you can also time lock it to sentence onset, because here it's time locked to picture onset, and these were auditory sentences, so they varied in length a bit. But if you time lock it to sentence onset, and this is all of the electrodes for all of the patients, you see that there's around 1 second, about the third word of the sentence, that's when, over the group, the theta power increases, or the difference between the two are above 0, right? And this I wasn't going to present really because it's not oscillations, but then given some of the discussion yesterday, I thought I might include it in here. So this is work that Anna did, Anna Jafarpour, maybe some of you know her, she's also in Bob's lab still. And she's interested in stimulus preactivation in the hippocampus. And she thought she had a nice way to actually test this with these data. So I'll try to reproduce what she did, which is too complicated for me to really explain thoroughly, but I'll try. So she used dynamic time warping, which allowed her to assess the similarity or the distance, it's actually the distance, but the similarity between two signals. And what she did was she compared, in this case, dissimilarity, because the distance, so the larger the distance, the more dissimilar they are, and that's why we are calling it the dissimilarity. She looked at the dissimilarity between, oh, sorry, maybe I forgot to explain this. So there's a baseline period, people hear the sentence, and then there's another 500 milliseconds before they finally see the picture, right? And they're supposed to name the picture only once they see it. So there's this interval here of 500 milliseconds when the context has happened, but now nothing is happening. And so what she did was to look at the similarity between this interval and that interval, and compare that to the similarity between this interval and the baseline, right? And the idea being that in here, if there's context and there's anything of a preactivation in the hippocampus at whatever level that is, this interval should be more similar to that relative to that. Is that clear? For the sentences that have a constraining context. And then the question that we're going to ask is, to what extent does this similarity predict the reaction times? The idea being, if you could actually preactivate anything in the hippocampus, whatever that is, that should make you faster once you see the picture to name it, okay? In the constraining context. And so this was done in the high gamma range, so 80 to 150, and that's why I don't think it has anything to do with oscillations, but I thought it's relevant given what Ole presented yesterday as well. So Anna came up with the pre-activation index, so just to make sure we are not modeling noise, the idea is to what extent this correlation between the similarity in here with the reaction times explains you anything above the correlation between this similarity and the reaction times. Okay, so just to normalize to anything that would be modeling noise. And this is what we find. So low here is the unconstrained context. High is what I'm calling the constraining context, so all of the sentences in the constraining context. And above 90% are the sentences in the constraining context for which more than 90% of the people filled in the sentences with the same final word. And so what we find is that if you would look at these two relative to when there's no context, the similarity in the signal between these two intervals is much better at predicting the reaction times for the picture naming only when there's a context in the sentence, okay? And so Anna takes this to be evidence for some type of preactivation in the hippocampus. We just don't know at what level that is. I don't think it's at the word level, but there's something conceptual going on. And so there's some signal in the hippocampus, or yeah, in this case, in the hippocampus, already prior to people seeing the picture, given the context that was going on. Is that more or less clear? Okay. So just very quickly, maybe what we're seeing in the hippocampus is the relating incoming words are being stored. They're stored in semantic in semantic memory. That's what the hippocampus is doing for language. I don't know really. I'm still trying to understand all of that. But for the purpose of this talk, maybe we could say that similar neuronal computations that are used by the hippocampus for memory function in general are also subserving ongoing language use in some way. And I don't know how much time I have. I think 10 minutes still. So actually this whole, the use of this task started a long time ago, not with the intracranial data, but actually with scalp data. So that's what I'll show you for the remainder of the talk. These are healthy university students, and it's the same task. Now they are seeing the words instead of hearing them. And just to show you, this is the whole reaction time distribution for the constrained condition and the unconstrained. So this means really, if you give people a context, what happens is that all of the responses get faster, right? And sometimes really by 200 milliseconds or so. So it's not something that, that you change the distribution. No, you're changing the whole, you're not changing the tail of the distribution. You make people faster in general. So it really suggests some type of priming, some type of, this context is really there's something going on that already gives you more of the response away of the picture concept. If you look over the scalp with EEG, this is constrained versus unconstrained for the different words. So at the beginning of the sentence, there isn't really much context, so there are no differences between the two conditions. But as you come closer to the final word, you start seeing at first these alpha power decreases prior to the next word. And then finally, when you're preparing or when you're given the last word and you're going to respond, you see it extends also more to the beta range, and it's more also more extended over time. Okay, so when you give people context, and I would say they can somehow prepare to speak, maybe not necessarily the phonological form that they are going to produce, but it goes with these alpha beta power decreases. Now, is this prediction or production? We're trying to find ways to test this. So one of the things that we can do is just have people look at the picture versus name the picture. If you do that, passive object viewing, you don't get anything. If you let people name the picture, then you again get to these alpha beta power decreases. And another way that we found to test it is by having people judge the picture with a button press with their left hand, right? So at least they're doing something, they are supposed to pay attention, but they are not overtly producing a word. They just have to say, is this the picture that I expected, yes or no? And so what you find, again, is the shift of the whole reaction time distribution. So people are faster in general once they are given the context, both for naming and judgment. And I'm now looking at this interval here, in this case, 800 milliseconds. This is naming, this is picture judgment for the theta, alpha, beta, and say high beta. This is 15 to 25 hertz. And so what we find again are these left lateralized power decreases in the alpha beta range when people can potentially retrieve a concept, even maybe retrieve a word and prepare to pronounce it. I don't know. If you localize the sources for this beta band, for the judgment task, they had to press with their left hand, so that was a good thing that I actually put whatever is going on in the judgment task in the other hemisphere. So you find there in the motor cortex power decreases. But for the naming task, what we see is this, it's pretty much the whole language production network showing these alpha beta power decreases. Yeah, so that's what I just said. And I think this is more than just prediction. There's more going on here than just prediction, because if you look at, when you let people predict but not name the picture, it's not exactly the same that is going on. If you compare this to the classic model of language production, from a meta-analysis in this case, I find it quite similar, and from this model I would then interpret whatever is going on in the temporal lobe as some memory process, so some retrieval from a concept or a word, and this frontal thing, the question is it prediction? We know that the LFG is supposed to be involved in prediction, so is this that we are picking up here prediction? Or if you would look at what Indefray and Levelt say, this here should be syllabification, so more of a motor thing rather than a prediction thing. So I don't know, one way that I've been trying to test it is to look at what happens if you have a lesion here, in this case a stroke-induced lesion versus a lesion there, okay? So just an example of a patient who has a frontal lesion and a patient who has a temporal lesion. And so these are patients, again, from Bob's lab. So I have 6 patients with temporal lesions and 5 patients with frontal lesions. This is the distribution of their lesions. So where you see red is where all of the patients in that group have, where their lesion overlap maximally. So if you have people, these patients and the controls do this task, again, you find this very strong effect for everyone. In this case here, I'm showing you the context effect itself, so by how much faster are people naming the picture if you give them context. And 0 here is no difference. And these are, again, their statistics for each individual, and you see that everyone shows these very strong context effects, except for 2 patients here, okay? 2 patients with temporal lobe lesions. If you look at the oscillations in these 2 groups, so these are all of the patients, these are all of the controls, it's noisier, the data are also noisier in general than what I had before, but it's still a very replicable effect. These alpha-beta power decreases prior to people seeing the picture is very, I find it over and over again. So these are the topographies for this alpha-beta power decreases for each one of the patients. And what I think you can notice is that P7 and P9, these are the 2 patients who are not really showing any of these alpha-beta power decreases, right? There isn't really much going on there, whereas all of the other patients have. It's very variable. It's also given their lesions, but these 2 patients don't show anything. So one of the things that I try to do is to see whether I can understand this more given the profile of their lesions. So I took the amount of damage there is to different areas that were relevant in this task. So the LIFG, middle temporal, superior temporal, angular, and supramarginal gyri. So all of those, the amount of damage to all of these areas, put it in a hierarchical clustering analysis, and what comes out is that patients P7 and P9 have a very different lesion profile from the rest. And I thought that was really, really cool, because these are also the 2 patients who are not showing the EEG effects and also not showing any of the behavioral facilitation effect, right? And you see here where what actually happens is that these patients have both a temporal but also an inferior parietal lesion, whereas the other patients have only frontal or only temporal or only frontal lesions. So here put together from this cluster, and this cluster, you see that P7 and P9 don't really show this effect very robustly. And then a final thing that this is something that Lars helped me do, that he did the very hard work for this particular study. So these are the 6 temporal patients that I showed you, that I just showed you. And we're actually going to focus a bit more on those now. So they did the same task. It's actually the same data that I just showed you. But I'm focusing here on the temporal patients exclusively. And when you look at only their data, not putting the frontal patients together, what I was really surprised to see, in the controls, again, you find this left lateralized alpha-beta power decreases. But once you see the patients, it seems to be going to the right hemisphere, right? At least from the scalp, you would say it's much more biased to the right than it was in the controls. And I thought, like, wow, this is really, really, really cool. So Lars did all of the source localization for these patients. So here you see where the lesions are. And then we tried to estimate the amount of damage that there was to the corpus callosum. So I didn't have tractography data on these patients. So what we were doing is actually looking at how many voxels were damaged in the corpus callosum. And then we're going to correlate that measure to the amount of power decreases we actually see in the right hemisphere. The idea being, maybe not all of the patients are showing these shifts to the right hemisphere because there's something with their corpus callosum, right? And lo and behold, this is just 6 patients. This is a non-parametric correlation. So most people say, like, it doesn't look like a correlation at all, but in this case, Spearman, so it's a monotonic. If one goes up, does the other one go up as well, right? And what we find is that only for these 2 that are reversed, otherwise it's a perfect, almost perfect relationship. So the more damage there is to the corpus callosum, the least we find in the right hemisphere, right? I'm really excited about these results. I don't really know yet what exactly, how it's going to the right hemisphere, why it's dependent on the corpus callosum, but that's the line of research that I'm going to pursue next. So I think, maybe, but I'll be bold and say, I think that this is, with these patient data, I can show a causal link between the alpha-beta power decreases and the context facilitation effect. And so that's one of the causal links, and the other one is to actually these posterior sources, right? So the patients who have a lesion in the temporal plus inferior parietal lobe are the ones who are not showing any facilitation effect, both in the EEG and in behavior. To me, it's still unclear what the role of the LIFG is in this effect. So Ole showed the data yesterday that clearly had this LIFG source in there, but if you lesion it, patients are fine, so I don't really know what's going on. And again, for the purpose of this talk, maybe what I'm mostly excited about, even if we don't understand exactly what these oscillations are, if we find them in the right hemisphere of these patients who have a left hemisphere lesion, potentially this means that the neuronal computations that the right is doing, that is helping these patients keep their language functioning despite the damage, are the same computations that the left would be doing. And that, to me, is the most exciting part of these studies. So, in conclusion, I think I have evidence, I will be very bold again, for the medial frontal theta in language production being related to resolving lexical competition. The medial temporal lobe theta, that is a memory signature in a language production task, relating incoming words to stored semantic knowledge. And finally, these alpha-beta power decreases in the lateral temporal cortex associated with maybe conceptual and or lexical retrieval. And maybe, so I think I believe, but I would like to believe that I'm on the right track of using these oscillations to find a way to bridge between language and other cognitive domains. And with that, I would like to thank you all and the patients who participated and my collaborators.
Thank you, Vitória. Excellent, talk. I have a question, for you regarding your, naming task, and I'm trying to understand how these relate to language as opposed to just activation of semantics. So, say, would you predict the same if you present a picture that has the concept, or if you present just a word? So, how much is this really language, as compared to just, something else?
Right. Well, but that's something else would still be semantic memory, then you're saying, right?
But then I wouldn't say necessarily language.
Right. So I don't know to what extent I can really say it's the one or the other, because I also don't make a very, in my head, there's not a clear distinction between semantic memory and language, or where does the one stop and the other one begin, so I'm not really sure. Maybe one of the things that I can say is that the patients who showed this impairment following the left hemisphere lesions, they do not have semantic aphasia, right? So there's nothing wrong with their semantics, and yet if you lesion, or given that lesion that they have, they don't show both the behavioral and the EEG effects. So that would allow me to say there's something really more language to it than just being semantic, right? Because then I would expect the patients also to have a semantic aphasia if it were the case that they couldn't do the task because of semantics.
But so have you tried, for instance, to do just 2 pictures? Have you tried, for instance, to do just 2 pictures as opposed to language?
So that's the thing. Yeah, that's the thing that I'm planning on doing. Okay. In September, there's a student coming, because also I want to be able to use this in the clinic and not all patients. I want to be able to predict who's going to use the right hemisphere or not. And not all patients can understand these sentences. So I'm actually trying to build a context just given pictures and have them still name or do something that is language related to the last one. And I'm hopeful I will find the same type of effect, but I totally agree with you. I don't know. But that's definitely on my plans. Yeah.
Thank you for the talk. I'd like to come back to your data on patients and...
Sorry, my data on?
On the patients with the left hemispheric lesions and on that shift of electrophysiological markers to the right hemisphere. So in the motor cortex, when you have a lesion in the motor system, when you see ipsilateral activation, it's actually generally a sign that the damage is more severe and that recovery is going to be worse. Did you actually correlate this lateralisation effect with the global severity of the aphasia?
Yes, it did not correlate with it. It did not correlate with lesion volume, which was also something to worry about. Maybe, it's just a matter of lesion volume. It did not correlate with that. It also did not correlate with the amount of lesion to particular areas in the left, so in the temporal lobe. The only thing it really correlated with was to the amount of damage to the corpus callosum. So that, yeah, it was really surprising at first, but it's only 6 patients, right? So it remains, there's really not much that we can say, but I think this is very promising.
the nature of the prediction. So I was kind of reminded of what Andrea said about whether the kind of N400 can go as far as predicting the phonological gestalt of the upcoming thing, right? So, because you were saying that maybe this IFG pattern that you see relates to syllabification, and isn't that kind of related to exactly that? I mean, how can it be if the prediction doesn't trickle down to the phonological level? You see what I mean?
Not really.
Okay, so if you predict the upcoming word based on lexical semantics, yeah, and you say the LIFG activity pattern that underlies this could indicate syllabification, and it would entail then that you predict something phonological from the lexical semantic context,
right.
and this is kind of against what you think, right?
I don't think that these data are about that, but I mean, I don't think we don't know, right? Yeah.
Yeah, so to me, this LIFG thing is still very puzzling, right? I'm not sure. I'm just borrowing from the model. It should be doing syllabification. And I know that some of the people in this task, there was a control who told me, like, do I really need Q8 for the picture? Sometimes I know what I want to say. I don't want Q8. I told him, no, you've got Q8, right? So it could be that they can already prepare a motor program to produce. But then again, then it shouldn't take them 600 milliseconds to speak, but rather 200 to 300 milliseconds. So that doesn't pan out either. So I'm not really sure what that LIFG thing is. But if you lesion it, then people can still predict. But they can still produce as well, even though at the level of the motor program, these patients may be a bit more impaired, which is known for these frontal lesions. So I really don't know.
Can I just quickly say back that I think there's a big difference between when you're preparing to speak and when you're passively either reading or hearing something, right? So if you are syllabifying something and passing activation down, it's a very different context.
So let's try this. This is the test for later on. But what might be going on in these power decreases that you see is the actual prediction of the actual word in one way or another, and you can directly test this. So you can see whether you can decode the content of a specific word from those squiggly lines, and I'd be very surprised if you couldn't.
Right. But then what is the difference? To me, what I don't understand is what is the difference between saying it's prediction, because you still have to say what the content of the prediction is. So to me, if you retrieve the word or the concept, why isn't that what I'm seeing rather than the prediction? Like, what is this prediction then, if not me being able to retrieve something? You see, that's where I think it becomes, the word prediction is just being used loosely without me being able to explain anything. Right, so is this
what you're doing is you're reinstating some kind of information using your memory, and that is what you see in the power decreases.
Right.
If that's what it is, you should be able to decode.
Right.
And as I will show later, from the power decreases, you can.
Right, but then it would be the retrieval. So these would be signatures of the retrieval and not of the prediction, which still remains an empty thing. If I just say these are signatures of the prediction. No,
What's the difference between retrieval and prediction?
Yeah, so to me, there isn't. To me, that's exactly the point. Exactly, but just some people don't. They say this is just prediction, and that remains to me very empty. Yeah, that's what I mean. But I'm really curious to see how you did it, so I'll try it.
Yeah.
I actually agree to be very careful about interpreting this as predictions, because you see these left lateralized decreases in alpha and beta also in cute overt reading, for example, before the sentence comes out, right? So there's no way to predict anything linguistic, just the motor preparation per se is sufficient to decrease alpha and beta in this entire network. So I agree with also the
Yeah, so I think that what's for me missing to have a more restrictive, narrow interpretation is either a processing model or more broadly, let's say, a theory of the task. That is what is actually a particular participant or patient doing moment by moment vis-a-vis whether this is a pair of pictures or a pair of words or actually a sentence, so that you can deconstruct moment by moment, because only then do we can't even say what the prediction is about. So. Other commentary on this?
But I also didn't embark on this thing because of the prediction. Like I don't.
Yeah, that's sort of it falls out. You see, we're all grabbing you on that, biting into your ankle and then holding on. Exactly. Okay, Oded, a short last intervention.
Yeah, well, I regret I didn't think about it before, but maybe in the time that we have before the end discussion, are we going to have an end discussion?
We are not going to have an end discussion.
Yeah. Okay, so then, because I thought maybe we should think of some definitions for prediction amongst ourselves, come up with some reasonable one, unified definition of what prediction is.
Well, yeah, I mean, we've gotten a few. I mean, Nancy put one up yesterday.
There was
a bit, and I encourage everybody to open a Google Doc to share with each other, and then have an exciting definitional competitions on it. But it's true that it's been confusing. Okay, thank you, Vitória. Let's move on to the next speaker.